Hello everyone, welcome to BioBoost 2025. Today's what we are going to cover is one another cell organelle which is Golgi complex. Beginning with the discovery of Golgi complex. It is noticeable with both light and electron microscope and is also known as Golgi body. The Golgi complex was first discovered by an Italian physician and Nobel laureate named as Camillo Golgi in 1898 during an investigation of the nervous system. Its electron microscopic structure was described by Dalton and Felix described its electron microscope structure in 1954. Location-wise, Golgi complex is present in all eukaryotic cells and absent in prokaryotes, meaning all the true nucleus having cells have a Golgi complex except the prokaryotes. The Golgi apparatus is especially extensive in the secretory cells because the primary function of Golgi apparatus is synthesis, so mainly its most majority is found in secretory cells. Secretory cells have majority of Golgi complex. It is absent in few cell types such as mammalian RBCs, sperm cells of bryophytes and pteridophytes, and sieve tubes of plants. A cell may have one large Golgi complex or several very small ones. It occupies different positions in different kinds of cells. Now we will describe the structure of Golgi complex. In this diagram here you can see a cell is present and Golgi apparatus here have a lumen inside and cisternae which are infolds which have two phases, that is the cis 1 and trans phase. So the different parts of Golgi apparatus, first is cisternae. So they vary from 3 to 7 in most animal cells, from 10 to 20 in plant cells. Cisternae may be flat but are often curved. The convex side of stack forms the cis phase. That is, convex side of Golgi stack has a forming cis phase, and concave side of stack has a maturing trans phase. Now secretory materials reach the Golgi complex from smooth endoplasmic reticulum by way of transport vesicles which bud off from SER, means smooth endoplasmic reticulum, and fuse with Golgi cisternae on the cis phase. How this process takes place we will see through a diagram. From the trans phase, secretory vesicles arises that carry the processed material to their destination. Means from here SER are present, this smooth endoplasmic reticulum here is present. Here through vesicles these are going to Golgi complex, and through that these vesicles go out or go to whatever destination is assigned to them. Next part of Golgi complex is tubules. Tubules are small round tubules arise from the periphery of the cisternae. Round and arise from the periphery of the cisternae. Third part of the complex comprises of vesicles. So vesicles lie near the ends, and concave surface of the Golgi complex, and concave surface of the Golgi complex. Concave means the trans face. Here you can see the trans face, and near to it these vesicles that you can see, outgoing transport vesicles, so these are present. Next we will discuss the functions of Golgi complex. Function wise, the major functions it performs include secretion, synthesis, sulfation, apoptosis, phosphorylation, and other cell specific functions that we will discuss in the last. Coming to the first function, that is secretion. Although the Golgi apparatus is involved in many different cell processes, its principal role in many cells is in secretion. In this diagram here you can see two types of secretion are there: constitutive secretion and regulated secretion. So we will discuss secretion part. It plays an important role in the synthesis of proteoglycans. Means synthesis of proteoglycans, which are molecules present in the extracellular matrix of animals. Here in this diagram you can see how Golgi apparatus is linking to ER, and through vesicles it is either sending these vesicles out of the cell and also inside of the cell through the different processes. One is showing exocytosis and another one is the process endocytosis. Next function, that is synthesis after secretion. Next function is synthesis. So it is a major site of carbohydrate synthesis. Means carbs are getting synthesized inside Golgi complexes. This includes the production of glycosaminoglycans, that is GAGs. These are known as GAGs in short. You can write them: glycosaminoglycans. Long unbranched polysaccharides, polysacs, are also getting synthesized in Golgi complexes, which the Golgi then attaches to a protein synthesized in the ER to form proteoglycans. So these together, Golgi send them to ER, from where they are made to proteoglycans. Because ER has ribosomes attached to it, so the ribosomal part, the rough endoplasmic reticulum, attach proteins to these. So when protein get attached to a carb commodity, then it becomes proteoglycans. Enzymes in the Golgi polymerize several of these GAGs via xylose link onto the core protein. So the Golgi apparatus is involved in cell secretion. Secretory vesicles are also shown here in this diagram, which break off the membranes and transport materials to the cell plasma membrane. So this is cytosol, this one is plasma membrane. Most important role that we see is of apoptosis. So it has a putative role in apoptosis, with several BCL2 family members localized there as well as to the mitochondria. Means along with BCL2 family members, it coordinates with the mitochondria to carry out the process of apoptosis. Next is the function of phosphorylation. So the phosphorylation of molecules require energy in the form of ATP, right? So that ATP is imported into the lumen of the Golgi, utilized by resident kinases such as casein kinase 1 and casein kinase 2. So because several molecules required phosphorylation, ATP that they need is imported into the lumen of Golgi, which then gets utilized by resident kinases present inside it. And we all know kinase is an enzyme which is responsible to phosphorylate any molecule it attaches to. So the kinases present inside the lumen of Golgi, examples are there, that is casein kinase 1 and casein kinase 2. One molecule that is phosphorylated in the Golgi is apolipoprotein. So one molecule that gets phosphorylated by some kinase is a protein named as apolipoprotein, which forms a molecule known as VLDL, that is a constituent of blood serum. It forms a molecule named as VLDL, which is a constituent of blood serum. Next function is of vesicular transport that Golgi plays. So basically, vesicles leaving RER. Here in this diagram you can see this is ER. So these are the vesicles that is leaving ER, gets transported to the cis phase. So basically this one is the cis phase, this one is the trans phase of Golgi. So vesicles that are going to the cis phase of Golgi complex fuse with the membrane and empty the contents into the lumen. Here inside this is lumen. So here these vesicles are going to Golgi and here they empty their contents. These molecules inside the lumen are modified and sorted for transport to the next destination. Here this is a modified vesicle which got modified inside ER. So basically it got modified inside ER, in Golgi, and they get sorted and sent to the next destination. Here because exocytosis is shown, so the vesicle is going directly outside the plasma membrane. Proteins destined for places other than ER and Golgi complexes moves to the trans phase. Means the proteins which are not meant to send to ER or Golgi apparatus, they move directly to the trans position. gets placed on either of the three vesicles, that is exocytotic, secretory and lysosomal vesicles. Here, because the vesicle which is getting transferred to the trans phase here, because the endocytosis is taking place, so the late endosome consisting this vesicle send it to the trans phase, and those particles which are meant to get degraded are sent to lysosome. So summarizing the concept, we can write that vesicles that are leaving RER, vesicles leaving RER transported to the cis phase of Golgi, fuse with its membrane and empty its contents inside Golgi's lumen. And molecules inside the lumen are modified and sorted for transport to the next destination. Proteins destined for places other than ER and GA, we know, get moved to the trans phase, and those proteins which are not meant to send either to ER or Golgi get transported to the trans phase of it and gets placed on either of the three vesicles that are either exocytotic vesicle or secretory vesicle or lysosomal vesicles. The other cell specific function that we discussed includes formation of cell wall in plant tissue is performed by Golgi. So basically cell wall formation, acrosome development in sperm, acrosome development in sperm cells, secretion of zymogen in the exocrine cells of pancreas, secretion and transformation of lipid in the liver cells. Similar secretory functions are carried out in the Brunner's glands, alveolar epithelium, Paneth cells and connective tissues as well. So, if we summarize Golgi complex, then we make points like it is a stack of membrane bound sacs, means it is a stack of membrane bound sacs. It receives most of the substances from ER. Receives most of the substances from ER. It produces vesicles which carry secretions onto the cell surface. It produces vesicles which carry secretions onto the cell surface. Basically, it functions as a post office of the cell where proteins and lipids are taken, labeled and transported to other locations within the body. means it basically attaches stamp to every protein and lipid which is getting taken, which is made and formed inside ER, and it transports to whatever destination they are supposed to go. So basically in this post office, proteins and lipids are taken, gets labeled and gets transported to their destined location within the body. So this was it about Golgi and what parts the whole complex has. In next video we will cover one more organelle. So thank you so much for watching.